Okay. So hello everyone. Uh, we are back to our session of genomics, and in which we were learning about transgenic animals and what is basically transgenesis and the concept of transgenesis is what we have seen in the last lecture. Like, what do you mean by transgenesis and how exactly you want to carry it out, and what do you mean by transgene? What do you mean by a transgenic animal? And that is what the introduction and little bit few examples we have seen in the last session. So the next thing that we have to we also learned the very important part of production of transgenic animals, which involves around 4 steps with the construction of a transgene. Step one is that. Step two is that introduction of foreign gene into the animal. Step three is screening for transgenic positives. Step four is further animal breeding is done to obtain the maximal expression in the animals. So those are the 4 steps, and how exactly you can integrate this particular gene of interest into an animal. Those 3 methods, which comes under the step two, that is introduction of foreign gene into the animal, which includes pronuclear microinjection method, embryonic stem cell method, and retrovirus mediated gene transfer. So out of which we had seen one of them, that is retrovirus mediated gene transfer method. So basically, we'll be using a virus as our vehicle to transfer that particular gene of interest into the animal, and we'll be making a construct and inserting that construct into the embryonic cells, 4 to 16 cell stage embryo, and then will be getting that particular animal once we hatch the means that this particular example in this they're talking about the chicken. How do they make a transgenic chicken? That is what they have shown over here. So that is what we have. This is what we have seen by far, and the very important step two, that is introduction of foreign gene into the animal, which has multiple procedures, multiple methods of conducting the same. We can use any one of the methods and conduct the experiment accordingly. Okay, but then retrovirus has its own disadvantages also, as in uh, the virus can also get multiplied in the host cell, and then it can show some virulent characteristics, and especially if you are going to um genetically modify that particular animal or organism for eating purposes, for food purposes, or anything that humans are going to use, then you might not want this particular method to be implemented to make a genetically modified organism. So that is the most important part, that disadvantage that we have with retrovirus mediated gene transfer. So later people concentrated on other 2 methods of transferring this transgene into the animals. So first one was microinjection method. So a female animal is superovulated and eggs are collected. So you do the ovulation. The female animal will be having undergoing the ovulation. So ovulation is nothing but the when the egg will be coming out of the this thing, the sac, and the fertilization sac basically, and then that ovule is collected. Okay, so female animal is super ovulated in the sense at the same at a time. If one egg can be made, means you can mm, do some injections, in the sense hormonal injection, and make that animal breed more than one egg. So at a time you can collect many eggs. So super ovulated and eggs are collected, then eggs are fertilized in vitro. Okay, so whatever that procedure of fertilization will be done in vitro. So the transgene containing solution injected into a male pronucleus. So you have female pronucleus, this one, and you have male pronucleus, and this is the egg basically. So male pronucleus is a part where the injection will be done of a transgene solution. Okay, so you can see a fine needle over here. That fine needle is inserting the transgene solution into the male pronucleus. Eggs with the transgene are kept overnight in an incubator to develop, and the eggs are then implanted into the uterus of pseudo-pregnant female. So what do you mean by pseudo-pregnant? She's not pregnant, but her body is prepared to be pregnant. In the sense, all the hormones will be secreted, and as soon as you put this fertilized egg, the body is ready to accept that egg and implant it into the uterus so that the further baby can grow over there. So that is what the pseudo pregnancy is. So the female is not actually pregnant, but her body is prepared to get the egg, fertilized egg, and attach it to the uterus, and where the baby can grow. So that is what it is. So micro injection technique is very simple. Most of the time people are using this particular type of technique. And female in very simple, the fertilization is done in vitro, in the sense you will be doing the fertilization outside of the body. And in this you will be injecting the solution that is containing your transgene into the male pronucleus. Okay, and then the fertilization of the egg will be happening, in the sense male and female nucleus will be combining with each other, and we have a egg, fertilized egg, and then that fertilized egg will be then again implanted into the pseudo pregnant female. And if I tell you, the same concept is used for doing what you say, IVF, in vitro fertilization method, when people want to have this method done. So same way things are done. The male pronucleus is taken, female pronucleus is taken. Maybe over there we are not inserting any transgene, but the in vitro fertilization is in vitro, and mother is pseudo pregnant, and then you will be putting the fertilized egg back into the woman's uterus and allowing it to get attached to the uterus so that the baby can grow over there. The same concept is there for in vitro fertilization as well. I'm just telling you it as a knowledge because it shares the same concept. The only thing we are doing over here, we are inserting the transgene with the help of a micro needle into the male pronucleus. So we are making sure that your transgene will be integrated within the fertilized egg itself. So somewhere in the fertilized egg, the gene will be integrated. Yes. Then after that is because of disadvantage of retroviral vector. Micro injection of DNA is currently the preferred method for producing transgenic mice. Okay. So because of disadvantage of retroviral vectors, with the main disadvantage of retroviral vector, I have told you already. This method of microinjection of DNA is currently the preferred method for producing transgenic mice, not mice. So the mouse was the first animal to undergo the successful gene transfer using DNA microinjection. This method involves transfer of desired gene constructed from another member in the same or different species into the proन्यूक्लियस ऑफ ए रिप्रोडक्टिव सेल. ठीक है, तुम अगर रिप्रोडक्टिव सेल के जीन को ही चेंज कर सकते हो या फिर ऐड कर सकते हो कोई जीन, then it will be much easier for you to produce a progeny which will be having that particular transgene, right? So then it will be cultured in vitro until it reaches the embryonic phase and then transferred to the recipient cell. So very simple technique, not very simple, but it requires lots of molecular diagnostic and molecular uh, preparation as well. And it takes a longer period of time. It doesn't take like 1 or 2 days to get the procedure done. Obviously, it takes around 2 to 3 months in order to get done with this procedure. And then we have animals or mice which with this that particular transgene integrated in their DNA. So very simple method in terms of understanding, and this is the diagrammatic representation of the same. So what does it have is inject the foreign DNA into one of the pronuclei. So you can either do it in male or you can either do it in the female. Then fertilized mouse egg prior to the fusion of the male and the female nucleus. So you have to do that before they are fertilized. So one of the nucleus will be putting the foreign DNA injection. Then transfer the injected egg into the foster mother. So pseudo-pregnant mother would can also be called as foster mother. So about 10 to 30% of the offspring separation uh, or offspring contain the injected foreign DNA. Foreign DNA is present in equal amounts in all the tissues, and mice expressing foreign DNA are bred to continue DNA in germline. So basically, once you are done with this, you will be getting, you will be injecting this particular fertilized egg into the mother, right? So then you will be getting, along with other babies, you will be getting what? Uh, 10 to 30% of the population which will be having this transgene. But then you want to breed this particular transgene containing animal only, containing my mouse only. So then you will be interbreeding them, you will be breeding them amongst themselves, or will be performing some breeding procedures which will give us all the mice who will be having this transgene in their germline. Okay, germline in the sense we generally call it as sex, what you say, reproductive cell lines. Okay, so that is what it is. So you have pronuclei, you will be taking one of the pronuclei, injecting with the transgene solution, then you'll be inserting that fertilized, then allowing them to fertilize with each other, the male and female pronuclei. Then fertilized egg will be then inserted into the mice. Then this mice will be giving birth to the another, means like mice, and one few of them will be having the transgene of interest. Then you have to take this particular animal who is having the transgene and bred them further to have all the animals who will be containing the transgene. So that is a simple procedure of microinjection of DNA into one of the pronuclei. Okay. Then embryonic stem cell method. So this is also one of the most commonly used method that we have. So in this, transgenic animals can be created by manipulating the embryonic stem cells. So now we will be integrating what? We'll be integrating the embryonic stem cells. So embryonic stem cells are obtained from the inner cell mass of the blastocytes. Okay, blastocytes is just one of the phases of growing of embryonic stem cells. Okay. So embryonic cells are obtained from inner cell mass of the blastocyte. Transgenic stem cells are grown in vitro. ठीक है. Transgenic stem cells means the one which we have already manipulated with our gene of interest. So you will be growing those cells in vitroli in your lab. Okay. Then they are inserted into a host uterus to grow normally. Okay. Then harvest the inner cell mass, that is ICM, from 3-day-old blastocytes, and then inject the transgenic embryonic cells into the blastocele of a new 3-day-old host blastocyst. So we'll see this in the diagrammatic form so that we'll understand it in a much better manner. Since it involves the embryonic stem cells, it is also talking about the various stages of development of embryonic stem cells. So you have a fertilized mouse egg. Then you have a targeted gene alteration made in the embryonic stem cells in the culture. So we'll be culturing them into the flask. Okay, that is what is shown over here. Then you have mutated stem cells injected into the blast cyst. So this fertilized egg will be getting converted into blastocyst. Okay. Then at this stage, we are going to inject this blastocyst with the stem cells which we have genetically transformed. Okay. And then allowing this blastocyst to get implanted into the foster mother. Then the offspring that you are going to get will be a chimera. Chimera in the sense it will be heterozygous in nature. Okay. it will be having some characteristics of this original mouse and some characteristics of this transgenic stem cell. Okay, then further breeding uh, for homozygous expression. So if you are getting this heterozygous mice, so let me explain this to you. So you have heterozygous mice, okay, and then you'll be having one more heterozygous mice, okay, then you'll be breeding them together, ठीक है, अगर मेंडेलियन जेनेटिक्स थोड़ा बहुत याद है तुम लोगों को तो at some stage I'll be still getting H heterozygous, I'll be getting one homozygous as well, right? I'll be getting two heterozygous, one homozygous. So I can get a combination of homozygous mice. So when I'm saying homozygous, that means homozygous for the expression that I'm looking for, the homozygous for the targeted gene alteration that I have done. Okay. So this chimera is basically heterozygous in nature. That means it has some part of this altered cells and some part of this normal mice. Okay? So I don't want this. I don't want this mice. I want this mice who is homozygous for the transgene that I have inserted. Okay. So very simple. So basically, you're modifying the uh, embryonic stem cells only. Okay, and then this embryonic stem cells you are inserting at certain phase, which is called as blastocyst. Okay, then this blastocyst will be implanted into the foster mother. Since we are inserting few cells, okay, over here at the stage of blastocyst, means developmental stage of embryonic stem cell, sorry, what you say, fertilized mouse, right? There are the I will be getting a chimera which will be heterozygous for the transgene expression. The transgene means जो जीन मैंने insert किया है वो उसके nature के लिए heterozygous रहेगा. So what I want homozygous transgene expressing mouse, right? So that means I'll be interbreeding this heterozygous mice together, and at certain stage, after one generation or two generation, I'll be getting a homozygous expression of my transgene in that mouse. Understanding this part, are we clear till here? Yes or no? Yes or no? Just tell me. So that was embryonic cell transfer. Okay. So we have learned three methods: retrovirus mediated cell transfer, transgenic mice method, transgenic production of animals, then microinjection, and also embryonic cell transfer. Then let's look into some example of transgenic animals, like people have successfully made some transgenic animals. So what are these animals are? So we'll see those. So transgenic animal में first है superfish. Uh, Growth hormone gene inserted into the fertilized egg, increased growth and size. Transgenic fish grows up to 10 to 11 times faster than that of the normal fish. So very important, very good. Then glowfish, genetically modified zebrafish, produced by integrating a fluorescent protein gene from jellyfish into the embryo of the fish. So they have basically combined the two characteristics, and then they have made a fish which will be glowing, right? So jellies are generally other fishes who glow. So they have added that particular gene into the fish so that we have a fish which can glow. Okay. So genetically modified, this is the regular salmon, and this is genetically modified salmon, which is super big in size. And this is the glowfish, which will glow, right? Then we have transgenic mouse. Okay. Then oncomouse. Okay, oncomouse, I have already told you about. Generally, we use nude mice to make oncogenic mice. Oncogene in the mice would be those mice in which we can easily have the cancer cells getting multiplied. Okay, So mouse model to study the cancer made by inserting activated oncogene. Okay. Then we have Alzheimer's mouse. In the brain of Alzheimer's patient, dead nerve cells are entangled in a protein called amyloid. So mouse made by introducing amyloid precursor gene into the fertilized egg of the mice. Then you will be getting a mouse which will be having this Alzheimer's disease already. Okay. So this is how they look. So this is what is Alzheimer's mouse, and this is what is mouse. You can see over here this particular uh, swelling that you can see is nothing but the oncogene, activated oncogene has been inserted and it has grown a tumor into the mouse. And this is how exactly the Moor mice look like. They don't have any hair, any fur on their body, and they are immunocompromised. Why? See, whenever I'll be injecting a foreign entity into my anybody's body, like any living organism, your own immune system will be acting, and your own immune system will be rejecting those cells, will be rejecting that particular antigen that is coming inside your body. So you want to make mouse in such a way that whenever I insert a oncogene, they should not reject that particular part. I cannot put probably at the embryonic stage for producing this oncomice. But if I have an immunocompromised mice already, it will be very easy for me to grow a tumor in mice body at any part. Okay. So generally, when I conducted this experiment in my lab, uh, what we used to do is like we used to just a minute actually. We used to make the solution of cancer cells, and we used to inject that cancer cell solution into their hind limb, this part. Okay. And if they have a very thin skin where you can easily pick up their skin and inject with a normal injection, and we will allow them to settle them over there and then see the growth of the tumor, and this cancer cells would grow over there very nicely, because these animals are what? They are immunologically compromised. Okay. So they don't have their own system so that they can accept anything that we insert in their body. Okay. So that is what it is. So these are just examples of few mouse models that people have created. Okay. And one is onco mouse, one is Alzheimer's mouse. Then transgenic pigs are also there. That is one of them is enviro pig and pig for organ transplant. So pigs have trouble fully digest in a compound known as a phytate in many cereal grains used to feed them. So transgenic pigs are created by introducing phytase gene of E. coli. Phytase enzyme is thus produced in the salivary gland of the pig, and it degrades the indigestible phytate in with the release of the phosphate that is readily digested by the pigs. So they have genetically modified this particular pig to make sure that they can digest that particular cereal grain. Okay. Pick for organ transplant. Pick with human gene in order to decrease the chance of organ rejection by human body. See, that is also very important. So whenever we have organ transplants done, okay, uh, so when you are performing that particular surgery, a doctor also has to make sure that particular organ is not rejected by the body. If that particular organ is going to get rejected by the body, then whole transplant procedure is a failure. So at that point also, we prescribe a medication to that particular human, that particular person who has got the transplant, which will be keep reducing their immune system until the organ is getting accepted by the body, and then they have to take that medication for the lifelong, understanding this part. So that many a times we say that the people who are having transplant or got done with the transplant, they have very low uh, power. That is only because of this reason why we have to prescribe them with some medication which will keep their immunity a little low, so that the organ which has been transplanted will be never rejected by the body. Okay. So whenever we are saying we are doing immunocompromised, we are making their body immunocompromised, or we are making immunocompromised animals, that means we are taking care that their antibody production against that organ is not happening. Okay. So when I was talking about these nude mice also, okay, maybe they don't have capacity to make antibody against whatever I insert in their body, right? So if antibodies are made against the antigen, then they make sure that the antibodies will make sure they're killing that particular antigen. So in immunocompromised systems, we are making sure that we are not letting the body produce any antibodies against that transplanted organ. Okay, so that is what it is. So the pigs were the one of the best animals that were used for doing some research on the organ transplant part. Okay, so this is how it is. Cute animal. Then transgenic sheep for good quality of the wool. We have already seen that. Then transgenic monkey. ANDI was the first transgenic monkey born in 2000. And ANDI proves that transgenic primates can be created and gene expression of a foreign gene delivered into their genome. I don't have much of an idea of this because I have never learned many things about it. But yeah, I have learned about mice and their characteristic and how we can use this transgenic animals to make to models for diseases. Then transgenic cow. Rosie was the first transgenic cow born in 1997 and produced human protein enriched milk at 2.4 gram per liter, contains human gene alpha-lactoalbumin. Okay, so this is how you can genetically modify a cow as well. Then recent research. So transgenic goats engineered to produce human breast milk. Okay. So University of California scientists created a transgenic goat by transferring human gene for breast milk enzymes and proteins into the goat embryo. Very, very edge, cutting edge research, I would say. Then produce 60% of lysosomes and lactoferrin found in the human mother's milk. For the babies of mothers who aren't present or can't nurse them, milk from this transgenic goat could be provided to provide the next best alternative. Okay, so this is what they have done. So making a transgenic animal, so you have human gene, you have egg donor, then you have micro injection of the human gene into the egg of this goat, and then development within a host goat, then transgenic goat, then milk from transgenic goat will be produced. Industry importance of this making these animals is toxicity sensitive transgenic animals to test chemicals. Many toxicity studies can be carried out. Spider silk in the milk of a goat. So I don't know what does this mean. Spider silk. Then medical importance, we will be concentrating on the medical importance. We can use it as a disease model, and we can also use for xenotransplantation and also to make the knockout mice. Just give me 1 second over here. Okay. So disease models, xeno, and also knockout mice can be made. What do you mean by knockout mice is that you don't want that particular gene to be present in the mouse. So you can make some genetic transformation. So you can delete that particular gene from the embryonic cells itself and then allow that mice to grow without that gene only. So that deleting of knocking out that particular gene can also make various changes in the mouse, and we can have, so if means if I tell you I don't want the, for example, I don't want P53 gene to be present in my mice. Okay, then I'll be doing what? I'll be making sure that I'm knocking out this particular gene from the embryo before it is before it is made into a egg. Okay, so that is what I'll be making sure. So see, this is how scientists have come to know that what all expression are, what are lethal genes and what are sorry, what are important genes and what are non-important genes, right? So when they went on conducting a few experiments in which they deleted few genes and then allowing the embryo to grow, and they later came to know, if I deleted this gene, the limb did not get produced. If I deleted this gene, then they had problem in the brain development. Some organ, vital organ development was hampered because of deletion of that particular gene. Then you would say that whatever the gene I have deleted is very important. Okay, and sometimes you would just allow them to breed amongst each other naturally and then later come to know about some kind of a disease that was prevailed because of some genetic combination. Then you will be like going back into this thing, and then you will be checking what exact combination of genes was done, so that we have gotten a, product uh, which is having some fault. Okay, so it has some particular term. It calls reverse mutation. Yeah, it is called, yes, forward mutation and the reverse mutation. So in the reverse one, you will be going back. In the forward one, you will be just allowing them to breed and check what effect it has. Okay. In the reverse one, you will be mutating few, and then you will be checking for the effect. So that is how you come to know which mutations are important and which mutations are non important. Okay. So sorry, not the mutation, which genes are important and which genes are not.